So amylin is hormone, pancreatic hormone that is synthesized and co-secreted with insulin by pancreatic beta cells. And the function of this hormone is to regulate satiation centrally. So it crosses from the blood to the brain. It crosses the blood to the brain in its normal state when it's a monomer. However, in many cases, amylin, as also beta amyloid, forms aggregates. So therefore, it's still floating around in the blood when it's aggregated and might not cross the blood-brain barrier as we anticipate and start blocking the vasculature. In fact, several years ago, we tested this in humans. We searched for histological evidence of amylin deposition in the brains in humans, and we found that amylin is deposited in microvasculature in the brain in humans that have Alzheimer's disease. However, we couldn't be able to find it in normal brains. So there is something that happened in individuals that have Alzheimer's disease that makes the amylin aggregated to deposit even at a higher level than compared with the normal brains. So there is evidence from histological studies in humans that amylin that is secreted from the pancreas is amyloidogenic and accumulates in the microvasculature in brains of individuals with Alzheimer's disease. There is a difference between the amylin from humans and amylin from other species, including the rodents. For instance, in rats and mice, amylin doesn't form amyloid, and therefore doesn't have the propensity to aggregate as the amylin in humans. So we took advantage, if I can say so, the difference between the human amylin and the rodent amylin, and made mice and rats which are transgenic for human amylin. We were very surprised the fact that in rats and mice that are transgenic for human amylin, the amylin forms aggregates as in humans and accumulates in the brains, and those rats and mice show dementia-like type of pathology and also change in the behavior function. So basically the rats that do not have Aβ pathology, so they are normal rats that transgenic for human amylin, they show deposition of amyloid in the microvasculature and show neurological deficits. Then if you cross the rats which are transgenic for human amylin with AD model mice or rats like APPS1, then you have the interaction between the amylin and the beta amyloid in the brain. And those rats, for instance, are normally showing behavior deficits at 16 months of age. However, when the amylin is from the pancreas, they show faster development of behavior deficits by 4 months. And also, we found in those brains mixed plaques between the amylin and beta amylin, showing that these two peptides do interact in vivo similarly as they do in humans. So the evidence behind the evidence that there is an interaction between the beta amyloid and the human amylin, I think it has been demonstrated from our lab and also several other labs that replicated our results, both in animal studies and also in human studies. So the fact that dementia is related to deposition of amyloid in the brain, and because the amylin from the pancreas interacts with beta amyloid, it suggests the hypothesis that if we are able to lower the amylin in the circulation in individuals that have Alzheimer-like dementia, it will slow down the progression of dementia. And we are doing this in animal models, showing that if you delete the amylin gene in normal rats, they even show development of behavior deficit even at a later stage. So even if it's not human amylin in the rats and mice that are prone to develop Aβ pathology, there is still a positive effect on the pathology and also on the brain function. Now, why the amylin from rats and mice still might be detrimental to those animals under conditions that they are prone to develop A-beta pathology, we do not know. But there are so many things that we don't know about this hormone. We envision the fact that individuals that have cognitive decline, the amylin that is in the circulation, even is not higher than in normal individuals, it's still a therapeutic target because it interacts with beta amyloid. So by preventing amyloid to deposit in the microvasculature and in the brain, we might be able to reduce the progression of Alzheimer's disease. So the next steps are to develop therapeutics targeting directly the amylin aggregated form in the circulation and reduce the deposition in the microvasculature, which will improve the brain health or prevent the brain microvasculature for being damaged, and therefore will help immunotherapy against the beta amyloid to be eliminated faster from the brain. So I think the amylin-centric therapeutics might prevent the development of dementia, might also improve the current therapeutics for directed against beta amyloid, and in the end, in individuals that have type 2 diabetes, it might be a way to reduce the diabetes complications in the brain.